In our previous class, we saw how life starts from this and ends in the infinitive complexity of adult human beings. In this class, we're going to be looking at something slightly different, and that is how cells die. And this is the perfect picture of a dying cell in mammalian organisms. Now, life from a mammalian cell can actually be seen as a continuous balancing act between the decision of remaining alive or going towards cell death. Most of the cells in our body remain quiescent, that is, they remain in the G0 stage of the cell cycle, which means that they are metabolically active, but they normally don't go through a cell cycle. However, they normally don't die. They remain active for a good amount of time. For a very long time sometimes, such as is the case for neurons. However, at some point some cells actually die, and cell death is something that is actually quite important for accomplishing the body plan that we have from the moment that we are conceived. So when you think of fetal development, a substantial amount of the processes that take place during fetal development are actually driven by some cells continuing to grow while some cells actually decide to go into apoptosis. And the best example of this is the cells that actually connect our digits when they are in the early stages of fetal development. At that time, our fingers are all connected, and the little thin membrane that connects our fingers actually has to be taken out so that the fingers can start looking as separate entities. And that separation is something that involves apoptosis, that is, cell death. So therefore, cell death is a part of the normal development pathway of most higher organisms, and it is something that is absolutely essential for morphogenesis and embryonic development. But that's not the only time when our cells can actually go into cell death. There are other times when going into apoptosis or cell death is the best thing to do. And one of those examples is during viral infection. When cells in our body are infected by viruses, one of the smartest ways in which our body protects itself is by allowing the cells that are infected to die quickly before the virus gets a chance to multiply within those cells. Another type of conditions that triggers cell death is when our cells are sustaining substantial amounts of stress, such as what happens during heat shock. And finally, another time when our cells decide to go into apoptosis is when they suffer too much DNA damage, the type that is considered to be non-fixable, that is, irreparable DNA damage. Now, it is important to keep in mind that cell death is something that can happen by different mechanisms. And while we tend to think of most cells dying apoptotically, apoptosis is not the only way in which our cells can actually die. There are two different ways in which our cells can die. One of them is by necrosis, and the other one is by apoptosis. Necrotic cell death is the one that is exemplified in the photograph on the left in this image, whereas apoptotic cell death is the one that is exemplified on the right. Now, the differences between apoptosis and necrosis are very, very remarkable. First of all, in necrosis, the cells actually trigger a pro-inflammatory type of signaling that leads to the production of a lot of different cytokines that lead to a swelling not only of the cell, but also of the tissue where the cells are located. From that point of view, necrosis can be seen as a process that is a lot more damaging for the body, because basically what happens is that the cell membrane explodes, therefore allowing for content that is normally contained in the inside of the cell to be exposed to the outside of the cell. And this is quite damaging for our organism simply because the exposure of intracellular content to the outside can actually trigger autoimmune responses. And autoimmune responses are at the heart of many human diseases. Now, on the other hand, apoptosis can be actually seen as the process in which the cell dies without releasing any of its intracellular components to the outside. So during apoptosis, there is chromatin condensation, DNA fragmentation, loss of membrane symmetry. But however, even though all of those things are happening, the proteins that are normally within the cell remain surrounded by membranes, and the DNA is not released out of the cell. Therefore, there is no chance for our immune system to start mounting responses against components that are normally located within the cell. One of the early events of apoptosis is DNA fragmentation, blebbing of the nucleus, and fragmentation of the cell. Now, right at the time when the nucleus of the cell starts falling into little vesicles, there is also loss of asymmetry in the plasma membrane. Keep in mind, the plasma membrane looks different from the outside than it does from the inside. In the inside, there are specific phospholipids, such as phosphatidylserine, that are normally not present ever at the outside of the cell. Phosphatidylserines are actually exposed to the outside of the cell during apoptotic cell death. The fragmentation that happens during apoptosis is in fact shown by the agarose gel that is shown in this slide on the right. This is what your DNA in a cell will look like. Time zero is normal DNA. Normal DNA is very large and therefore it doesn't run too much in an agarose gel. This is the normal status of your chromosomes. However, when the cell goes into apoptosis, that DNA starts getting cleaved, and at 3 hours, the cleavage of that DNA has already generated a ladder that is clearly visible in this gel. Now, by 12 hours, the cell has gone well into apoptosis. Most of the cells have actually gone into apoptosis if you provide a apoptotic stimulus to the cells. And now you can clearly see the kind of DNA fragmentation that is achieved when your DNA is cleaved in between nucleosomes. So remember, this profile resembles very much the profile that we saw during cleavage of the chromosomal DNA by micrococcal nuclease. So this is what happens at the plasma membrane. This is the normal asymmetry of phospholipids in the plasma membrane, and this asymmetry is actually lost during apoptosis, and it's one of the early markers of apoptosis. And the main phospholipid that I want you to remember is phosphatidylserine, which is normally facing the cytosol, and during apoptosis is then translocated to the outside of the plasma membrane. Now, I want you to pay attention to this movie.
Or apoptosis has been induced in these cultured cells. Cell death is characterized by blebbing of the plasma membrane and fragmentation of the nuclei. Suddenly, cells lose all attachment to the substratum that they have been growing on and shrivel up without lysing. The mechanism of apoptosis involves many tightly controlled steps, 3 of which are visualized here by different staining techniques. One initial event is the sudden release of cytochrome C from mitochondria into the cytosol. This event has been visualized here using fluorescently labeled cytochrome C. Initially, the green-yellow staining is restricted to a reticular pattern, which then suddenly disperses as the mitochondria release their content proteins into the cytosol. At a later step, the lipid asymmetry of the plasma membrane breaks down. In normal cells, phosphatidylserine is only found on the cytosolic side of the plasma membrane, but when cells undergo apoptosis, it becomes exposed on the outside of the cell. This event has been visualized here by adding a red fluorescent protein to the media, which specifically binds to phosphatidylserine head groups as they become exposed. In an intact organism, exposure of phosphatidylserine on the cell surface labels the dead cell and its remnants to be rapidly consumed by other cells, such as macrophages. Finally, although apoptosing cells don't lyse, their plasma membranes do become permeable to small molecules. This event has been visualized here by adding a dye to the media that fluoresces blue when it can enter cells and bind to DNA. All 3 of these events can be observed simultaneously.
Apoptosis can be triggered by two different mechanisms. One of them uses signals that are coming from the inside of the cell, and a second one is activated by signals that are coming from the outside of the cell. The one that is triggered by signals coming from the inside is considered to be the intrinsic pathway of apoptosis, and the one that is triggered by signals coming from the outside of the cell is considered to be the extrinsic pathway. Among the type of stimuli that will trigger the intrinsic pathway are radiation, toxins, and exposure to free radicals. Basically, events that have the ability to damage the DNA of the cell. On the other hand, among the ways to activate the extrinsic pathway is exposure to specific ligands that have the ability to activate receptors that then go on to activate cell death. Among those receptors are the FAST receptor and the TNF receptor.
When a cell is under stress, such as from DNA damage, a viral infection, or growth factor deprivation, it may undergo programmed cell death, also called apoptosis. Such death signals are routed through the mitochondria in a series of events known as the intrinsic pathway of apoptosis. Death signals within the cell trigger the activation of pro-apoptotic proteins such as Bax. When activated, Bax forms oligomers in the outer membranes of mitochondria. Formation of the oligomers leads to the release of cytochrome C from the mitochondrial intermembrane space, either by forming pores or by interacting with other mitochondrial outer membrane proteins. While in the mitochondrion, cytochrome C participates in cellular respiration, but in the cytosol, cytochrome C drives the cell into apoptosis. Cytochrome C binds to the cytosolic proteins called APAF1. With cytochrome C attached, the APAF1 proteins can assemble into a structure called the apoptosome. Another protein, called Caspase 9, is now activated by forming a complex with APAF1 in the apoptosome. Caspases are proteases that have cysteine residues at their active sites and cleave after aspartic acid residues in their substrate proteins. Two types of caspases play roles in apoptosis: initiator and effector caspases. Initiator caspases, such as Caspase 9, are the first caspases activated by apoptotic signals. The function of initiator caspases is to activate effector caspases, such as procaspase 3, through proteolytic cleavage. Caspase 9 cleaves procaspase 3, producing active caspase 3. Caspases, like other enzymes, act catalytically, allowing a small number of initiator caspases to activate a large number of effector caspases. The accidental activation of caspases could result in apoptosis, but this is prevented by the presence of a family of proteins called IAP, for inhibitor of apoptosis. Members of the IAP family directly interact with caspases and suppress apoptosis by either inhibiting caspase activity or by targeting caspases for degradation. In mammalian cells, the permeabilization of mitochondria by Bax results not only in the release of cytochrome C, but also of IAP inhibitors. These inhibitors block the action of IAPs, allowing the cascade, beginning with cytochrome C, to activate caspases and culminate in apoptosis. The effector caspases are sometimes called executioner caspases because they are responsible for digesting more than 100 different target proteins, thereby mediating the events of apoptosis. One key target of the caspases is an inhibitor of a DNase, which when activated is responsible for fragmentation of nuclear DNA. In addition, caspases cleave nuclear lamins, leading to fragmentation of the nucleus. They cleave cytoskeletal proteins, leading to disruption of the cytoskeleton, membrane blebbing, and cell fragmentation. They also cleave Golgi matrix proteins, leading to fragmentation of the Golgi apparatus.